Oh, okay, everybody. Do we have any questions before we get started today? No? Okay. Well, then we'll get started right off the bat. I am going to put in the discussion here a worksheet that when you're done, you can start practicing or working on that. Let's see here. I keep getting an error message when trying to download. Okay. So here's the file for the worksheet. And it is a PDF, so you can hopefully write over it if you have an iPad or something. Go ahead and do that. But that's the worksheet we'll be working on in our small groups, okay? So let's get started here. I'm going to post the passcode in the chat here, and you guys can get started. Good luck, everybody. Oh, I'm not sure why you guys aren't seeing me. I just realized that. There we go. Lucky you, huh? Here we go. Journey, please see the message I sent you. Roshan, please see the message I sent you. Journey, please lower your camera. All right, everybody, come on back. Get Journey in here again. Okay. All right, let's start our PowerPoint. I just need to send one thing here.
Sorry, Miss E. This was my distraction. So when you see
Oh,
the
awesome.
distraction, so I'm so sorry.
That's all right.
Here.
Okay, here we go. All right, you guys. So there are a few students who were not able to take the exam, so we just need to hold off, and then during remediation, if you would like to go over it, we can do that. Okay. All right. Let me just pull up my PowerPoint. All right. Let's go here and so today we're shifting gears because we are now going to more of the histology portion of the course, histology and embryology. So most of you guys have already taken a, can those of you who are here, can you guys please type your name in again? Because I want to take attendance of who's staying after the exams, okay? So many of you guys have already, or you should have already taken a biology course, right? So what is it, what do you guys remember about cells? And since there's not very many of us, I'm going to go ahead and unmute everybody so we can have kind of a discussion here. Let's see here. Participants. You know what, it's not going to let me. So go ahead and just press your space bar and tell me what you guys remember about the cells.
The mitochondria is the powerhouse of the cell.
That's right. That's what gives it the energy. That's what gives us energy, right? Okay. What else do you guys remember about cell development? Or cells, I should say, in general. Nothing? All those biology courses?
There are two types of cell, prokaryotic and eukaryotic.
Okay.
These consist of the DNA or the RNA or the genetic material. And then there's the endoplasmic reticulum that's responsible for the production of proteins.
Very good. Yep. Very, very good. All right. How about the rest of you guys? Anything else? No? Okay. Well, let's have a little review here, and hopefully you guys all got that worksheet. Did you all get the worksheet off of the chat? Okay, great. All right. Well, let's start here. I'm not sure which screen it's sharing. Okay. All right, so by the end of this first part, you should be able to describe the cell and the function of its components. The cell is basically just a bag. I think of it as a balloon full of fluid. And then the balloon, the outside of the balloon is the cell membrane. And that's what keeps the fluid in and the foreign materials out. Okay, so when we say foreign materials out, what does that mean to you guys?
Bacteria.
Yeah, so that's one. Very good. Bacteria. Okay. Now, some molecules can pass through. What molecules, like just off the top of your head, what do you think can pass through a cell?
Water.
Water. Depends on the type of cell membrane.
Oxygen
Oxygen.
and lipids.
Yeah, lipids. How about medications? Okay. I haven't had enough coffee yet today, so I'll be drinking it here throughout. Cytoplasm. That is the fluid found within the cell. Okay, so that's our water in the balloon. The nucleus is then found in that cytoplasm, and the nucleus contains DNA and RNA, and they control all of the cell's operation. So tell me what you guys know about the difference between DNA and RNA.
DNA has 2 strands, RNA has 1 strand.
Okay. Anything else? So what does that tell you if you have 2 strands versus 1 strand?
DNA can split to replicate.
Okay, very good. All right. So let's go into it here on our next slide. So the DNA is found in the chromosomes. All right, so these are the ones that are going to give us our characteristics. Okay, the RNA is found in the nucleus, and it carries the genetic information from the DNA to the part that makes more cells that's going to replicate. That's mitosis. Okay, so usually only 1 nucleus per cell in the RNA, and at times there may be more than 1, but we aren't going to worry too much about that. And this picture here should look familiar if you guys looked at the worksheet at all. So we have many parts of the cell. Notice this. Let me just spotlight. Okay, notice this pinocytotic vessel. What do you guys think that does? Notice how it kind of invaginates there. What do you think that's doing?
This the foreign bodies or anything that can come into the bacteria, it engulfs it and then digests it.
Very good. Very, very good. So that's part of our phagocytosis, right? Yeah, I'm just going to leave it there, not get too overwhelming here. Here's our nucleus, and what does that do? That's the what of the cell? You all have very big ones. That's the brains. Okay, that's the brains of the cell. All right, all of this kind of beige color is the cytoplasm. We have our lysosomes, which are important with amino acids, and we're going to get a little more into that, I think, a little later on here. There we go. So organelles, that is most of the cell parts. They're small functioning parts, and they allow the cell to remain alive and to carry out its function. So here's our organelle, or one of them, and this is the mitochondria. And what did Kinza, or was it Kinza? Who talked about the mitochondria? Was it you, Chelsea? Somebody
No,
did.
it wasn't. But I can't see your screen. It says started screen sharing. It's just black. I don't know if anybody else sees that.
Is anybody else having problems with that? No? You can all see my screen, right? Yes? Yes, okay. So I'm not sure what's happening, Chelsea.
Dominic replied in the group and said he can't see it either.
Oh, okay. I'm not sure. Did you guys do your Zoom updates? That might be part of it.
Maybe I'll go log out and then come back in and see if that helps.
Okay, sounds good. All right, so our mitochondria is responsible for all the energy production. And so this is part of our metabolism. And we're going to talk a lot about that in pharmacology, the metabolism. And can somebody tell me what you guys know about metabolism? What does that mean? I mean, it does say it on here.
Breakdown of food particles to release energy for the body.
Yeah, okay. So and then inside, we see all these little cristae, and they are folds, and they're created by the inner membrane. So when we see more cristae, we know that there's more cellular action. Okay, everybody write that down. More activity. Cristae, increase in cristae equals more activity.
Christe? Oh, that's how you say it? Christe?
Cristae.
Christe.
And they have the enzymes that are going to help with metabolism. So the more they increase, the more you're going to burn, right? The more energy is going to be produced. Okay, now let's talk about the endoplasmic reticulum. And this is interconnecting tubules within the cell fluid. And if you guys printed out that form, well, let's go back a few slides here. Okay, so we have a couple different types of endoplasmic reticulum here, right? We have rough and we have smooth, but I don't think they show it on here. Okay, rough, what makes it rough is that it has proteins on its surface. Oh, let me get Dominic in here. So it has the ribosomes on its surface. Sorry. And that's what makes it rough. I always think of like little speed bumps all over it. Okay. Sorry, having to enter people in here. So that is going to produce proteins. And then the smooth endoplasmic reticulum, it doesn't have any ribosomes on it. There we go. Then we have our Golgi apparatus, and this is our packaging center, okay? So it packages the protein secretions and it moves it around the cell and then outside of the cell without mixing with the cytoplasm, okay? So think of this as Amazon, right? They're packaging everything and they're sending it out, maybe keeping some things in. Then we have our merocrine secretion, and that's the Golgi apparatus produces a thin membrane to surround the protein. That's kind of the package. And the protein then moves to the inner cell surface, or to the inner surface of the cell membrane, and it fuses. Okay, so it's just sticking on that cell membrane. Then when the cell ruptures, the membrane is then going to release it. Now we have our lysosomes. This should just be a refresher. Our lysosomes, and they are the scavenger for the cell. So what does a scavenger do, you guys?
They look for treasures.
They look for treasures, right? So they're going to go and they're going to clean everything up. I always think of them as a little vacuum. They're cleaning everything up and digesting the foreign bodies and other organelles that die, okay? These are contain digestive enzymes, and if injured, enzymes can leak out and consume the cell. So what gives the cell its shape? The membrane, but then sometimes we have things on that membrane, right? And what might we have on a cell membrane?
Is it a phospholipid biolayer?
Yes.
Isn't that what it's made up of?
Wow, impressive. Phospholipid bilayer. But what these microtubules and these filaments that hold its shape, they're hollow rods. The microtubules are hollow rods, and you guys are going to hear about that when we talk about enamel also, hollow rods. And subunits of the protein tubulin. The microfilaments are rods found on, oh, make sure you guys circle this. Okay, circle that. Microfilaments are solid round rods found in all cells except mature erythrocytes. What are erythrocytes? Erythro means what?
Red blood cells?
Red. Erythro means red, sites for cells. Okay? So they're found on all cells except for mature red blood cells. Then we have our centrioles, and those are bundles of these microtubules functioning in mitosis by aiding in the alignment of the poles and dividing the cells. Okay? So they just help divide the cells. What does inclusion mean to you guys?
Spillways.
What's that?
Spillways.
Spillways?
In the mouth. Yeah.
Okay. Those are embrasures. But what are inclusions? Well, it was close. Started with you know, we don't judge, right? Somebody tried. All right. What are inclusions? Cell inclusions. What do you think that's going to do?
Like union of different types of cells?
Yep, so it's going to kind of, yeah, a union, I guess. It's more, inclusion means like including everything. So it's not produced by the cell, but it's stored for use elsewhere, okay? So it's including, so if you go to an all-inclusive resort, everything's included, right? So it's included in there, and then but it's going to be used somewhere else. Maybe a lipid droplet or glycogen. What does lipid mean?
It's a fat cell.
Okay, and glycogen, what are we thinking there?
Sugar.
Sugar, carbohydrates, right? So both are forms of energy when required to be released to travel where they need. So inclusions enter by pinocytosis. You guys remember that? We just showed that. It looked like that invagination again, where it's that phagocytosis. It's part of that. Bringing it in, it caves in and pinches itself off. So that means a drinking in, and you guys want to know that, okay? So right, pinocytosis, a drinking in.
How I remembered it is like Pinot Noir, like the wine.
Good one. Who said that?
Kelsey.
So yes, a Pinot Noir, and they're drinking it in, okay? Very good. All right, now let's go to the epithelial tissue and the glands. So we want to be able to define the function of the epithelium and name the various types and their location, to describe the origin of glands and the ways in which they may be classified. So you guys tell me what you know about glands so far.
They secrete hormones.
They can secrete hormones. Very good. What else can they secrete?
Enzymes.
Enzymes. Okay, what about, what are the two different kinds?
The adrenal glands.
So that is, what type of gland is the adrenal gland?
The two types are endocrine and exocrine.
Very good. Endocrine meaning what?
Ending in ducts.
So endocrine, it means it's staying in the body. And exocrine means it's going outside the body. So what do you think a gland is that drains outside that drains outside of the body? That we
Sweat
might, as
gland?
Okay,
hygienists,
sweat glands. Oh, saliva gland, the salivary glands.
very good. Salivary glands. Okay, those are excreting into our mouth. And as Samantha said, there are enzymes in there that are going to be, and minerals that are going to be beneficial to fighting disease, fighting cavities, helping remineralize. So very important function. All right. So here we go. Epithelial tissues. This should be just a review. It's a group of cells that make up skin and line the inside of the tubes and cavities of the body, such as our glands, our pancreas, and liver, all originate from epithelium. Has different classifications according to number of cell layers. We have our single cell layered or our simple epithelium. You guys remembering this from biology? Simple and stratified, right? And then our multi-layered, which is stratified epithelium. Simple epithelium or simple squamous epithelium, they're going to be flat or plate-like, right? They're just going to have one layer, not very protective if it only has one thin layer, right? Remember, what are the sizes of some of these cells? Do you guys remember from the beginning? They're really, really small, right? Like microns, okay? And this allows for the exchange of oxygen and carbon dioxide between the linings of the lungs and the capillaries. So that's where a thin layer would be really important, right? There are two types. We have our endothelial cells. So that means inside. And that's going to line our blood vessels and our lymphatic vessels and the heart, things that we need, that exchange of oxygen and carbon dioxide. Then we have our mesothelial cells, and that lines the body cavities. I always think of kind of middle when I think of meso. All right. Then we have our simple cuboidal epithelium, and that is, self-explanatory. You guys should know that by now. Simple meaning one, cuboidal meaning cubes. This is located in the kidneys, glands, and respiratory passages. There may be cilia present to trap contaminants and move them out of the respiratory tract. So what happens to a smoker? What happens to these cells?
They burn off.
They burn off, but they also, the cilia, as they build cilia, get trapped in that tar of the smoking and aren't able to move things away as easily. Okay, but yes, they do die off, and they don't work as effectively prior to that. Okay. Simple epithelium, columnar. We know columnar, they look like columns, right? They're going to absorb products within the digestive tract. Again, we have these microvilli, which are these nice little structures here to help break things down. They're also found in glandular ducts. Okay, so we have ducts in our mouth, right? So we know that we have some simple columnar epithelium in our oral cavity. Pyramidal cells, so columnar cells that are packed together to form small ducts. And then we have the basal end. When you see the word basal, think basement, think bottom, okay? And that's the side facing the underlying tissue. So it's going to be connected by the basement membrane right here. And then here's our connective tissue. So our epithelium is more outward than our connective tissue. The apical end is the side facing the free surface of the lumen. Lumen meaning hole, the free surface or lumen, okay? So pseudostratified, remember it looks, does this ring a bell to you guys from biology? Pseudostratified, it's fake. Every time you see pseudo, it's like pretending to be something that it isn't. It looks like it's stacked, but it's not, okay? The goblet cell is a single cell gland intermixed in the epithelium. So these often have goblet cells, okay? Or are goblet cells. So these secrete mucus and lubricate the epithelial surface. So here's what it looks like. It looks like they're stacked, but when we look at them closer, we see, oh, they do all touch the basement membrane, okay? Cilia are our small hair-like projections of the epithelium, just like the microvilli, only they're a little bit longer. They're going to trap contaminants and move them to the nasal passages or the oral cavity to get them outside of the body, okay? Then now we're moving into our stratified, our multi-layers. So stratified cuboidal or stratified columnar epithelium, not commonly found in the oral cavity. 2 or more rows of cuboidal or columnar cells on top of one another, generally only found forming large ducts of glands, okay? Our transitional epithelium, what would you think that is? Transitional, what do you think it's going to do? What does transitional mean, you guys? I'm trying to get you guys to key in on these certain words so that when you get to your test, you can say, oh, I know that word, instead of, oh no, I don't know this answer. You can start thinking it through, okay?
It's going to change?
It's going to change, right? So it changes in thickness and in appearance as the need arises, okay? So this is found in the urinary system, and everybody circle that, know that transitional epithelium is found in the urinary system. Why do you think the urinary system would need that? Why do you think your bladder needs transitional epithelium?
To expand.
Yes, the bladder's going to expand, which is going to make it thinner, and then it's going to contract, making it thicker. Okay. All right, stratified squamous epithelium. No, your boards may ask you this. It is the most common type of epithelium. When you think of the oral cavity, you're going to think stratified squamous epithelium. So this makes up the mucosa of the oral cavity, the pharynx, the esophagus, and the anal region. Okay, so stratified squamous epithelium, that is the majority of the oral cavity. That is our buccal and labial mucosa, okay? Now, it's going to have, we're going to have several layers here. And one thing I want you guys to know, I don't know if it asks in this class, but this will come up a couple times throughout your time here. You'll be tested when we test you to kind of remember what you've learned in previous classes. The stratum corneum. Does anybody know what the stratum corneum is? Or what it does?
Does it like stretch? It's the most superficial layer.
It can stretch, yep, depending on where it's at. But what it does is it gives it its waterproofing. Okay, so when you see stratum corneum, think of the outer layer of your hands, and it can kind of be thick. Sometimes it gets keratinized, which means thickened, okay? And that makes it kind of waterproof. So when we wash our hands, the part that keeps the water from all going into our body is that stratum corneum, okay? And so you guys can see all the different layers. When I see stratum spinosum, know that the stratum spinosum gets kind of shaped like a star. And so I always think, and that's kind of, that can often be a question. So if you mix those first 4 letters, it spells star. So those are irregular shaped cells that end up looking like little stars. They have like little spikes on them sometimes, okay? And then the stratum basale is always going to be lined with that basement membrane. And we have the basement membrane is generally a desmosome. So desmosome meaning it's like double-sided tape, okay? It's going to stick to the cells of the epithelium, and it's going to stick to the cells of the connective tissue. Sometimes we have a hemidesmosome that only has one-sided tape, okay, where it's only sticking to one side. All right, so let's go through these layers. Our stratum basale, again, it's going to be cuboidal cells resting along the basement membrane right before it goes into that connective tissue. Layers where cells divide and form more cells to replace the cells that are lost. So our skin is constantly renewing, and this is constantly shedding, okay? The stratum spinosum, again, it's pushed out from the stratum basale layer and named stratum spinosum because they have the little spikes on them. Stratum granulosum, this is where we start to see evidence in thick skin, or we're starting to see this in thick skin, and it appears as 2 layers or 3 layers of flattened cells. So things can start changing. If anybody here ever go like this when they're testing, do a lip bite, all right? So you might have more layers because it's calloused there, all right? And that is keratinized, all right? Then our stratum corneum resembles a fingernail tissue, but it's a lot softer. 3 different layers are present. You can read through those. Stratum lucidum, this is thick skin, such as the palm of your hands and the soles of your feet. So generally, we aren't going to see this in the oral cavity, but this lies between the stratum granulosum and the stratum corneum. So the stratum corneum is still on the outside. All right, there's our picture again. What time are we looking at? I think we're okay. All right, epithelial tissue pigmentation. Do you guys remember talking about melanin and melanocytes in your biology course? Gives us the pigmentation, and sometimes we'll see that in the oral cavity. We'll see different types of pigmentation, which is different than disease. So what would be the difference in colors between those produced by melanin and those produced by disease? What might the tissue look like with melanin and the mucosa? Go for it, Neil.
I just don't know.
Okay. Zoe, what might tissue in the mouth look like if it has melanin?
Like what kind of color would it be?
Yeah.
Like darker, like more like a brownish color?
Very good. Very good. More like a brownish color. And Brittany, what might it look like if it's infected? What color would we see?
Like more red?
More red. Very good. So knowing the different types of colors and what causes them is important to know. Okay, so we have our melanin and our melanocytes that are going to give us a darker color. So we're going to see that more in darker colored skinned people. Okay. Now our glands are mostly developed from epithelium. As the epithelium develops from the basal cells, it grows downward into the connective tissue. So a little bit different, right? Forms a cord of epithelial cells that's hollow, with a hollow out to form a tube. The tubes reach a certain depth in a bulb-like or tube-like process and forms on the ends. These little forms on the ends are acini, I believe it's acini, or tubules. I've heard it pronounced too many different ways. So you guys can see here where it comes down into the connective tissue. The epithelial cells are coming down into it. And then you get these little grape leaves here, okay? So they're classified by distribution, what they secrete, the arrangement of the components, the products, and their embryonic origin. So how do they distribute? They produce, the products they produce are carried away from the gland. So the exocrine glands, as we talked about, are going to secrete via ducts to the outside of the body. Endocrine glands have no ducts. You guys need to know that. Endocrine glands have no ducts. And products are carried away in the bloodstream. So Gao, was it you that said it secretes hormones? Who was it that said that? Somebody said that. And so the hormones are going into the bloodstream, right? Okay. I think Zoe needs a break here. I saw that. All right. So how do they secrete? Let's see here. The holocrine gland is when the entire cell dies, and it's secreted and expelled when the cell membrane breaks. Where the merocrine gland, and we're going to be talking about the merocrine gland a little bit more as we go into our gland sections and chapters. And this, the secretions pass through the cell wall, allowing any cell cytoplasm to escape. Don't worry about this right now. I'm just introducing you guys to this because we're going to be talking about it later on, okay? Exocrine glands, again, are going to secrete fluid, and it's a modified substance. What do you think is secreted in saliva, you guys? What do you think these glands are secreting? What do you think is in saliva?
Enzymes?
Enzymes. What else? What minerals do we need to rebuild our enamel? Do you guys remember what our enamel, well, we haven't gone over it too much yet. So calcium. Okay. And when you drink water, fluoridated water in the city, you're going to also excrete it through your salivary glands. Okay. There's more, and we'll talk about that later on. So we have simple tubular, which is a straight tube, and then compound, which is numerous levels of branching within the duct system. So here we have our 3 different kinds of secretion by the salivary gland. So we have serous, which is thin, watery, and contains mostly digestive enzymes found in the saliva. So this is going to help break your food down. All right, so serous secretions help break food down. Then we have our mucus secretions, and those are going to be a little bit thicker. Okay, and why do you think we would need something a little more thicker, a little more viscous?
For like a coating of the mouth.
Perfect, very good. For a coating in the mouth. And then we have seromucus secretions, and that contains both serous and mucus cells in varying quantities within the same gland. All right. I think we should just take like a 5-minute break because I know that this is a lot of information, a lot of recall. So I know your brains are probably exploding right now after a test and then this. So go ahead and take just a really quick 5-minute break and then come back and we're going to talk about the embryonic origin. All right, you guys, come on back. Okay, let's go back to our PowerPoint. All right, so now we're going to talk a little bit about the embryonic origin, and we are going to start with, right off the bat, the egg and the sperm meet. They are now creating the embryo. Okay, so it starts off as a blastocyst, and I love that it's a blastocyst, because when I think of cysts, I think of fluid filled sacs. Okay, and so this is a hollow ball formed shortly after fertilization. The outer layer, so it starts first with 1 cell, 2 cell layers, 3 cell layers. When it gets to 4, it starts to form this sac, okay? And the outer layer is the outer, oh, this must be on a timer. I hope it's not. This outer layer forms the membrane surrounding the embryo and the placenta. And then the inner layer of the cell mass, this part here, is going to produce the embryo, or the embryo and one membrane layer, the amnion. If you guys remember the amniotic fluid, okay, that's all derived from this. Then we get this primitive streak that then goes downward into the middle of the embryo. The inner layer initially has 2 layers of epithelial cells, an ectoderm and an endoderm. Okay, ecto meaning outer, endo always meaning inner. Later, we get this middle layer that's called the mesoderm. So the ectoderm is going to develop into the epidermis and the CNS, the central nervous system. And then the endoderm is going to form the epithelium of the digestive tract, respiratory, and some glands. So endo, in, we're getting our digestive tract, our respiratory system, and some glands. Our mesoderm, or mesoderm, depends on who's lecturing, forms connective tissue, bone, cartilage, squamous cell lining of the abdomen and the thoracic cavities, and the inner lining of the blood cells. Okay, so that's kind of in the middle of the digestive tract and the epidermis, right? Or the respiratory surface in the epidermis. All right, let's talk a little bit about our connective tissue. So at the end of this, you should be able to describe the component, function, and location of general connective tissues, to briefly describe the structure of bone and the 2 ways in which it's formed, and to briefly describe the components and origin of blood cells and their function in normal numbers. So we know that this goes under the epidermis, right? We have the basement membrane. Now we're going into that connective tissue. So all types originate from the mesoderm. Connective tissue can be divided into connective tissue proper and then specialized connective tissue. That's going to be our cartilage, our bone, and blood. Okay, and you can kind of see that those are specialized, right? Those aren't our norm. All right, so it is composed of cells, fibers, ground substance, extracellular fluid, and it's subdivided into dense and loose connective tissue. So our connective tissue proper is going to be our dense connective tissue. There's 2 types, always, right? Regular and irregular. It does include both, our dense regular connective tissue is going to include tendons, collagen fibers, and those are the ones that are going to run parallel to each other. Fibroblasts are squeezed between them. What are fibroblasts?
Those are the ones that creates fibers.
Yes, those are our cells that are producing fibrin. Okay. So those are squeezed in between, and those are what's going to keep regenerating and keep creating the fibers in our body. So this allows tendons or ligaments to repair themselves if they aren't completely torn. If it's just a slight tear, then they can regenerate that tissue. Now, our dense irregular connective tissue is named because the fibers are going to run in all different directions. So regular runs parallel. It's source of nutrients it is a source of nutrients for the skin. It contains cells, fibers, ground substance, nerves, and blood vessels. Fibroblasts, connective tissue cell that forms all collagen fibers. So think of, when you're thinking of collagen, you need to think of how our teeth, what's in our gingiva, okay? What's being destroyed in our gingiva during periodontal disease? That's why it's important for us to know about fibroblasts and collagen fibers. Different medications are going to help aid in the reproduction of this, of collagen, okay? Now, our dense irregular connective tissue is non-elastic. It holds epithelium to muscles or bones and holds bones together. It attaches muscle to bone and holds the teeth in its sockets. Okay, we don't want elastic when we're talking about our teeth being held into the sockets, right? So we're going to look at our dense irregular connective tissue. They are mesenchymal cells. And one thing that's interesting about mesenchymal cells is that they are often stem cells. And they are now finding mesenchymal cells in teeth that aren't fully formed. So what do you think the benefit of that would be? Look at what it produces. Why do you think these stem cells would be important?
You could grow more tissue, possibly.
Yeah, yep. And look at the types of tissue, you guys. Bone, cartilage, muscle, fibroblasts. That's huge, right? I do think that this is the future. You know, when you guys have patients that are going to have teeth removed, I believe in the future there's going to be an option for them to bank those cells. So you think of deciduous teeth. So my daughter has a condition where if this were to happen, some of the cells actually in her teeth might actually help, even though they're mesenchymal, with some of the tumors in her brain. Okay. But look at this, bone production. What if somebody loses a leg? Do you think we could create a bone for them? Think of all of our soldiers, right? Cartilage, muscle, fibroblasts. These may be grown in a lab later on, or maybe put into into somebody's body, and they find their way to differentiate and create this. Pretty amazing, right? The science, it's being worked on as we speak. Like that, well, I don't know with COVID if they're still in the labs, but this is where research is going. So when I do think in probably within 10 years, you're going to have to give your patient an option to bank those cells if this keeps going the way that they're anticipating. It's very interesting. Also creating new teeth. You know, maybe somebody was born with, just a couple teeth, right? Maybe we can help grow them, right? So instead of implants, which have their own issues. All right, so anyway, this allows for the replacement of components of connective tissue lost through injury. Tissue lymphocytes, cells in connective tissue produce antibodies, virtually the same as a lymphocyte in blood. So we have our T lymphocytes and our B lymphocytes. Lymphocytes originate from the bone marrow, then pass to the lymphoid organs, where they then multiply. Okay. T lymphocytes are the most their most common function is to combat virus infected cells. Okay. It also helps combat tumors and grafted tissue and organs. So we need these to enable us to do a graft. So you think of somebody has recession, they need a graft. We need to have these T lymphocytes to help fight anything that may be preventing that graft tissue from attaching. All right. And you can see in the next line, if these T lymphocytes, they must be destroyed or suppressed in the transplant patients, or they'll reject the transplant or the graft. So think of burn victims, right? So when you guys are hygienists, and you have a patient who maybe just had a, we'll say, kidney transplant, what kind of medications do you think that they would be on, knowing what we know?
Immunosuppressants.
Immunosuppressants. Yes. We want to lower their immune system so it doesn't go after that organ. Okay. So how do you think, if we're lowering somebody's immune system, how do you think that would hinder periodontal, our treatment of periodontal disease?
Can the bacteria like penetrate easier then?
Well, they don't penetrate easier, but they don't get destroyed once they penetrate. Okay, so they're allowed to grow more. And that's what the problem is with immunosuppressed patients. Okay, is that we have to try and fight that infection for them. So maybe they need to come in more often to prevent the furthering of their disease, or them from even getting the disease. All right, so then we have macrophages, and they act as scavengers. Again, they're going to clean up all those dead cells and anything that doesn't belong in there. Then we have our adipocyte, or our fat cell, that's found in the bottom layer of the connective tissue. Fibers present in connective tissue, we have reticular fibers. They function as a framework for the organs. And then we have elastic fibers, and those stretch and return to its original length. So as we get older, you see those elastic fibers, those are in our face. So those elastic fibers, the reason we get wrinkles is because those elastic fibers lose their ability to return to their original length. Okay, and that's why we get wrinkles. Then we have ground substance, and that is glue-like substance that holds everything together and holds the cells and the fibers together. All right, let's move on to our loose connective tissue that's found in superficial and deep fascia. What are fascia? Some believe fascia should be an organ.
Skin.
What is it?
The skin.
They're more like spaces in between layers. Okay. It supports the organ's internal framework, and it's made up of collagen, elastic fibers, fibroblasts, macrophages, and mast cells. Mast cells play a big part in allergies, so we're going to be talking about that in pharmacology. I'm trying to tie this in to let you guys know the importance of understanding this. Connective tissue proper, loose connective tissue with special properties. We have our mucous connective tissue, primarily made up of fibroblasts and collagen. This is found in the umbilical cord and in the vocal cords. Then we have elastic tissue. This is made up of fibroblasts and yellow elastic fibers. It's found around the vocal cords, supporting tissues, ligaments in the spinous process. Then we have our reticular tissue, and that's the framework for most organs. We have our adipose tissue that's composed of fat cells. Remember, that's at the bottom layer of our connective tissue. Then we have pigment tissues, that's our melanocytes or melanoblasts, and they produce melanin granules that increase the skin's pigmentation. I don't have many, as you can see. All right. Cartilage. Cartilage is a connective tissue. It's not calcified. That's what differentiates it from bone, is that it's not calcified. Okay. These are chondroblasts and chondrocytes, fibers of either collagen or elastin, and you're going to find ground substance in there. There's 3 types. We have our hyaline cartilage, our elastic, and our fibrous. Our hyaline cartilage is the firmest type of cartilage. Okay. This is what allows the arms and legs to lengthen. Okay. Why we can do this, right? It's seen in adults in the larynx, trachea, bronchi, nasal cartilage, and certain parts of bones. So think of your nasal cartilage. It's pretty firm, right? Then we have our elastic, which is very flexible and is found more in the ear, the epiglottis, and the eustachian tube within your ear. Then we have our fibrous cartilage, and again, that's going to be mainly fibers, and it functions as a cushioning substance. So this is what we're going to find in our temporomandibular joint. All right. It's just this fibrous disc that's going to help cushion our TMJ. All right. So it grows in 2 ways. It grows by apposition and by interstitial growth. Apposition meaning it's layer upon layer upon layer, right? Then we have interstitial, and that's where it's undergoing cell division and enlargement within. So instead of growing layer and layer and layer, it's growing within here, which is making it bigger. Okay. Then we have our interstitial growth is how long bones will grow. So once somebody stops growing, that's done. Okay. Spongy bone, that is the bone found in the middle of the bone. It's also known as bone marrow. And you'll see small bridges of bone with spaces between. And when we start looking at radiographs, you guys are going to see like these little pockets, and that's going to be our spongy or cancellous bone. Then we have compact or dense bone. That's going to be our outer hard layer of the bone, and that's going to be made up of our osteoblasts, because that's what's creating bone, or osteocytes, collagen fibers, and then hydroxyapatite. I want you guys to make a note of hydroxyapatite, because that is also what we find in teeth. So anything that's good for teeth is going to be good for your bones. Okay. So it is a microscopic crystals of calcium and phosphates. Now, bone is about 50% mineralized, and that's going to become more important later on when we start talking about how hard enamel is. Do you guys think enamel is, we already did talk about that. Enamel is harder or softer than bones?
Harder?
Harder?
Enamel is harder because remember, it's 96% mineralized.
It's the second hardest organic compound behind diamonds.
Yeah. Yep. Okay, so very, very hard. So without hydroxyapatite, bones would be flexible, okay, without that mineralization. Intramembranous formation, it's formed within the tissue. Primitive mesenchymal cells become osteoblasts. They're then surrounded by the periosteum. The outer layer of the periosteum is collagen. The inner layer is osteogenic cells. It's just a layer outside of the bone. The cells then secrete the ground substance, collagen fibers, and hydroxyapatite. Osteoblasts get trapped and become osteocytes, and those are the cells that are embedded in the bone. Let's see here. Let's see here. I'm just trying to find. Here we go. The epiphyseal plate is between the epiphysis and the diaphysis of long bone, and that's where the growth happens. So how many of you guys have broken a bone and they say, oh, at least it wasn't the growth plate when you were younger? Anybody ever heard that? No. Okay. My childhood may have been a little more adventurous. So when somebody's younger, they want to make sure that that growth plate hasn't been disturbed. If that breaks, then that's when they need pins and rods to help that bone grow. Okay? So when the pituitary gland stops producing growth hormones, that plate disappears. That's why we don't have to worry about it as an adult. And that's when our growth, how tall we are, it's going to stop. Okay? So again, bone is about 50%, hydroxyapatite crystals, so 50% mineralized. The rest is collagen, ground substance, and water. The outer surface is the periosteum. Inner wall of the marrow is lined with endosteum. Endosteum forms modified bone on the inside during remodeling of bone and bone growth. All right, let's move on to our cortical bone or plate. It is a hard structure between the periosteum and the marrow cavity. So when we talk about bone plates, there are different types. Like when we do an implant, if it disturbs the buccal plate, it's not going to be successful. Buccal, remember, meaning outside. But for some reason, it can disturb other, the lingual plate. This has numerous blood vessels running through it, has osteocytes, and then the Haversian system, and that is just the blood vessels you're going to see on the next one running through here. So here are these, the Volkmann's canals. All right, and it grows in, it looks like a tree, right? In these different circles. If you cut a tree down, you're going to see different stages of life. Bone is kind of the same way, and that's why they can tell if a bone's been broken. So nutrients pass through the osteocytes, veins carry blood and new cells back to the bone, out of the bone back to the heart. You guys should know this. Interstitial lamellae are between the Haversian system, that's the rings of bone. Okay, we're going to talk about those when it comes to teeth also. All right, let's talk about blood. We know it's fluid and it's full of cellular parts. The fluid is plasma. The cellular is our red blood cells, our white blood cells, and our platelets. Erythrocytes, erythro meaning red, no nucleus found. It is a biconcave disc. Have you guys seen that under microscope, or it kind of dips in? All right. This contains the hemoglobin, which carries the oxygen from the lungs to the cells, and it also carries the carbon dioxide from from the cells to the lungs for elimination. So in anemia, we're going to see a decrease in the number of red blood cells. Leukocytes are white blood cells. We have two groups. We have granulocytes and agranulocytes. Granulocytes have granules of the cytoplasm. We have in the granulocyte category, we have neutrophils, eosinophils, and basophils, and then agranulocytes, we're going to have lymphocytes and monocytes. So the granulocytes function as phagocytes, killing and devouring any toxins, whether it's dead cells or anything that's come in there that doesn't belong in there. And these are what's going to cause an inflammatory response. So when you think of when gums start to turn red and swollen, this is what's happening. Those granulocytes are going in there to devour and take out that bacteria. But when it's too much, that's when we get that inflammation. So 4 signs of inflammation are redness, warmth, swelling, and pain. So those are all things we need to key into. Increase in fluid is plasma that leaks from the smallest blood vessel. Fluid contains neutrophils, which begin to attack the microorganisms. Here we have our eosinophils, and they help combat allergic reactions. All right, so now we're talking about those immunoglobulins, right? And they fight parasitic infections. Then we have our basophils, and those along with mast cells. Remember I said we're going to talk about allergies next semester and containing those that mast cell production. So they do contain histamine. Histamine is released, and that's what causes that reaction, that allergic reaction. All right, agranulocytes found in the circulating blood and the lymphoid tissue. Okay, 2 types, we already know this, B and T. They provide immunity. So when we talk about immunosuppressants, it's going to be affecting these B type and T type cells, our agranulocyte lymphocytes. Then we have our monocytes, and these become macrophages in acute inflammation, so a sudden inflammation. During disease process, the number of white blood cells increases to stop the infection, right, to combat the disease. In leukemia, you're going to see too many white blood cells develop and mutate and choke out the red blood cells. So we need to key in on this, because as hygienists, if we see more white blood cells, more bleeding that can't be explained any other way, we need to refer our patient to their physician. I know at least once, I feel like twice, I've referred my patient to their physician, they had leukemia. Okay, so we need to key into this and know, hey, they didn't bleed like this last time. There isn't any more calc. There isn't any other disease going on here. So we need to send them to their physician to get some blood work done. Do we tell them it might be leukemia? No. Okay, we're not doctors, but we are very well-educated dental hygienists, and we know that if we see too many coming to the area, we need to refer them. Same thing with a patient that has leukemia. I've had several, and their physician always says, if they start bleeding beyond their normal, you need to halt the cleaning and send them back to their physician for testing, because that could be one of the first signs that they're out of remission. Okay, everybody cool?
Would you then, I have a question about that. Would you then tell like the doctor that you kind of noticed that they had more bleeding, and then the doctor would then talk to them?
So at Herzing and in a lot of offices, you'll give an oral assessment report, and you would say, gingiva bleeding beyond expectation, please run blood tests, or you can't really order them, but I always say, you know, please evaluate for physical. And then they always draw blood at the physical. Usually these patients haven't been into their physician. I always ask the patients, you know, how long has it been since you've had a physical? Oh, the last time I saw my my dermatologist. No, I want to know when the last time was you had blood work drawn to check all of your levels. Okay, because that gives me a really good indication that if I see a lot of bleeding, then I need to refer them. Okay?
Is that a conversation that you would need to have with the dentist first before you bring it up to the patient?
I would not bring up leukemia to the patient.
No, but just if you are advising them to go see their doctor.
That depends on your relationship with your doctor and your experience. I honestly, my doctor usually asked me because I've always loved pharmacology and anatomy and how the body works. So he trusted me with that information. But usually when they come in for the exam, just say, you know, I saw more bleeding than usual. So I've referred them to their physician. And, you know, sometimes it's diabetes, sometimes it's leukemia, you know, it could be a plethora of conditions. But the fact is that that patient hasn't been into their physician recently and they need to be evaluated because something's going on. Good question, Samantha. All right. Particles of a larger cell called megakaryocyte are found in the bone marrow, and this is under platelets. Platelets have an important role in clotting blood. They release serotonin, which we're going to talk about a lot in pharmacology, causing blood vessels to contract. Other platelets stack up in leakage areas and form a blood clot. So platelets are very important. You're going to find that there's a category of antiplatelet medications, okay, because they are clotting too much, right? All right, everybody stretch. How are we doing on time? For some reason, okay, 11:23. I do have a video for chapter 18 and I think 19 that were found in the from last year that I did post. So you guys should be able to go through that, okay? All right, let's go on to our muscles and neurons, and I want the students to discuss the 3 types of muscles and their functions, shape, and locations, and discuss the neurons and its parts and function. So we know we have 3 types of muscle. You guys remember this from A&P, right? Skeletal, cardiac, and smooth. So our skeletal tissue is going to be that's connected to the bones by tendons. They're made up of thousands of myofibers, which is muscle cells. So these help with the contraction, with the control of how you move, okay? They are striated and voluntary. Your brain can tell them what to do. I'm telling my arm to move right now, so there we go. This is all skeletal muscle, okay? We have the myofilaments of actin and myosin. The actin filaments slightly overlap the myosin filaments, and a chemical reaction causes the 2 filaments to slide over one another. And then we have a sarcomere, which is a functional unit of the skeletal muscle. It should all be review. Our cardiac muscle, again, is involuntary because it's cardiac. We don't tell our heart to beat, right? It is not under the willful control. Sometimes we probably wish it would be because then, you know, we could tell it to beat when it stops, right? 1 or 2 nuclei per cell. Muscle cells or fibers branch as they meet one another. Again, the biggest thing with this is knowing that these are involuntary muscles and that the Purkinje fibers are specialized muscle cells in the heart that act like nerves and conduct messaging through the heart. So those are fibers that are communicating with each other. that's why they're involuntary. All right, then we have smooth muscle. This is our non-striated involuntary muscle. No stripes, and it's not under willful control, found in the lining of the digestive tract. So while it's not under control of us, it's of the autonomic nervous system, right? It's managed automatically. So it's found in the lining of the digestive tract, the blood vessels, and the lungs, and many other organs. Pretty much everything that is part of the autonomic nervous system that isn't the heart. Our nerve tissue is the communicating system of the body, sensory or afferent messages. Sensory meaning you feel it and it's carried from there to the brain. Motor, the brain is going to it to tell it what to do. All right, so there's three parts of the neuron cell. We have the cell body, the axon, and the dendrite, and they carry messages by sodium and potassium ions. We're going to talk about that a lot when you guys get into pain management and give local anesthetic, because you need to know what's happening there. So the passage of an electrical charge to the brain, which then converts it and interprets what it's saying and then sends the message out. You guys should know most of this. You guys can read through it. All right, I want to talk a little bit about the myelin sheath, because I talk about that a lot when you guys get into preclinic. In preclinic, so you guys are all very bright and you've gotten to preclinic because you're so smart, and now all of a sudden your brain isn't what you need. Now you need more muscle and muscle memory. And the more that you use muscles, it creates a sheath over the nerve to help it tell it what it needs to do. Okay? So it's a protective covering around the axons. The size of the axon and degree of the myelinization control the speed of message to which the nerve travels. So you're going to be able to automatically tell your hand and your fingers what to do. At first it's very awkward, but the more you do the motion, the faster that message is going to get to the hand. It plays a role in the regeneration of damaged nerves. If the myelin sheath remains intact, the nerve can regenerate until it reaches the point where it once supplied sensation. If the myelin sheath is damaged, it might not reach the same place. All right. Not sure how this prefacial embryology, these kind of got out of order, it looks like. Okay, so this is chapter 18 or 19, excuse me, I think. You know, I'm going to repost this because it looks like these got out of place. I was wondering why we were talking about that earlier. All right, so next we're starting the prefacial embryology, and I'm going to redo this, resubmit it to you guys. All the slides are there. But we start going into the development of us and how we develop. And I'm going to send a couple videos out. YouTube has a lot of really great resources if you have problems understanding the development. But things that I would like you guys to know is that the stomadeum is the future mouth, okay? That the first pharyngeal arch is the mandibular arch. That's important to know. The second, the mandibular process also forms the maxilla. The mandibular arch also forms the maxilla. And then cleft lip, you guys got to know about cleft lip and cleft palate when they happen. And know that the maxilla closes in a backward movement like a zipper. So it's zipping back. All right, so the earlier that the cleft happens, the more open it's going to be. Okay. So here it's talking about the cleft palate occurs between 7 and 11 weeks. Circle that. Right now it's slide 91. Giving you guys a second for that. And then the closer, remember it's a zipper, so the closer to week 11, only the soft palate or uvula will be affected. So if it's at week 7, it's going to be affected more anterior all the way back. But each week it's zipping just a little bit more as it goes back. Okay. There it talks about clefting. This is all on that video. Okay. All right, I think that's about it. I'll send out a couple YouTube suggestions that I have for the embryonic stages, and then you guys can go from there. Any questions on today? It's a lot of information, but a lot of it should have just been review. Nobody? You guys look exhausted. It's a lot on a Monday morning, isn't it? All right, you guys. I'll let you go a little early since we went straight through, okay? Remember, chapter 19, I believe, has a video. If it doesn't, I'll create one, okay? All right.
Can we just stay on here for remediation?
No, I have to do it on a different video. Anybody else? Who all plans on going to remediation? Okay, a couple of you, 3 of you. Okay, great. All right, you guys, if you have any questions, please feel free to email me. Yes, Esther?
We can't talk about this test on remediation, right?
As long as nobody is in there that still has to take it, we can talk about it.
Okay.
So, all right. And that's why I'm going into the different, well, I'm supposed to go into the other video anyway. All right. Kate, every time I see you, I think you could be related to me with that red hair. A lot of people in my family have that same color.
I'm the only one right now.
Oh, really?
Well, my cousins do, but they're starting to go blonde.
Yeah, I'm getting a little blonde here, which is like supposed to be our version of gray as a redhead. You turn blonde, I guess. That's what they say. All right, Zoe, do you have any questions? Oh, Zoe was leaving. All right. Oh, no, you're right there. I'm trying to find you. Okay.
No, I'm good.
Okay. How about you, Ashley? I saw you sent me a message. I'll get back to you, okay? All right.
I have
Anybody
some
else?
questions about the worksheet that you gave us.
So we'll do that in remediation.
Okay. Yep, that's all.
Okay. All right. Everybody good? All right. I'm going to start the remediation, okay?